So today is Tuesday, September 28th, 2021. We are going to begin a very heavy week of discussing the endocrine system. I know with lecture, I've got endocrine system and lab, I've got histology for the endocrine system, but really we're going to fold the two into each other. And as we work through each gland, we will talk about the histology associated with it, because I think that's the most poignant way to do that. Hopefully you have your lists of the hormones and also the histology you're responsible for, because like I said, our goal will be to go through that together. One more assignment due in this section, well, really two. The first is your Labster, which is due on Thursday this week. Remember, it must be 100% complete and 80% correct for full credit. And remember, you can complete it multiple times to make sure that you reach that goal. The second homework thing you have is the group presentations. Again, the presentations are going to be next Tuesday, 1 week from today. However, this Friday, by 6 p.m., your outlines are due. Again, I can't emphasize this enough. I don't like having assignments due on days when we're not in class because I find too many people forget about them. So don't make that mistake. Make sure you turn in your outlines on time or early. As soon as you're done with them, go ahead and turn them in. The assignment page is set up there for you. Because it is vitally important that you turn them in so that I can get them posted onto Canvas site so people can start looking at them. If your outline is late or if you do not turn in an outline, you're losing 10 points. And remember, this is a 50-point assignment. So that's 20% of your grade right off the top if you're not turning that in on time or if you turn it in at 6:03. So make sure it is turned in on time. I tend to look the other way on 5-minute delays on people turning in their homework assignments at the beginning of the class, but that will not be the case here. This is a hard deadline. Late assignments will be penalized heavily. For these, remember, only one per group is necessary. Make sure everybody's name in your group is on there, your topic is on there, and also make sure you have your references on there as well. And like I said, next week is party week for me. I get to sit back and take the week off because on Tuesday, you guys get to entertain me with your group presentations. And on Thursday, you get to entertain me with the lab and lecture exams. So I get to just wear my Hawaiian shirts, drink my Mai Tais, and enjoy next week while you guys do all the heavy lifting. All right. Questions on any of that? All right, excellent. Then let us dive in. As I mentioned, we are on to the endocrine system. I think this is one of the more challenging organ systems for a couple of reasons. For starters, unlike the cardiovascular system where you can hold the heart in your hand or the skeletal system where you can hold the femur in your hand, there isn't a whole lot to hold in your hand with the endocrine system. I mean, yes, we have glands like the thyroid gland, the pituitary gland, but just holding a gland and look at it doesn't tell you what it does. The endocrine system is all about chemical signals, histology, and interactions of those chemical signals with cells and the effects that they have on the cells. So that makes it probably one of the least intuitive sections. And as I was telling someone earlier in office, in my office hours, this typically is one of the more challenging sections of this class. The first section is somewhat challenging, especially for those who didn't have me for 430 because they aren't always aware of what to expect from something like this. So there's a wide diversity of scores, but the same is true for this section as well, because this one is kind of the least intuitive and the most challenging. But as I also warned that student, while this one may be the toughest section, it definitely gets easier from here on out. So after you survive this section, the last three are just a gravy train. So it does get better. Now, hopefully you recall from 430, one of the big things we talked about in 430 were exocrine glands. And what's the big difference between an exocrine gland and an endocrine gland?
Endocrine goes into a duct.
Which one goes into a duct?
Exocrine?
Yeah, exocrine, absolutely. These use ducts. right? Oops, that doesn't need to be capital. Right. Exocrine glands use ducts, and of course they use those ducts, as the name indicates, exocrine. Crine means to secrete. Exo means what?
Outside.
Yeah. So these use ducts to secrete substances outside of the body. And give me some examples of some exocrine glands.
Sweat glands,
Sweat glands, excellent. Give me one more.
sebaceous glands,
Yeah, sebaceous glands, excellent. Any gland not in the skin? Those are both perfectly acceptable, 100% correct answers. Any others?
Do tear ducts count?
Yep, tears count. Lacrimal glands absolutely count. What about salivary glands? Do salivary glands count?
yes.
Yeah, absolutely. Again, remember the lumen of our digestive system is outside of the body. So any gland releasing into our digestive system is also an exocrine gland. Mammary glands, excellent. All great examples, absolutely. Right, this is different from endo, within, so to secrete within. These secrete instead of outside the body, inside the body. And in most cases, that is into the interstitial fluid. Of course, once you're in the interstitial fluid, as we learned about in the first section of this class, where do things go from the interstitial fluid?
The lymph nodes?
I'm sorry?
Blood.
Yeah, into the blood. And of course, once it's into the blood, where can it go?
Throughout the body.
Yeah, exactly. Everywhere. Exactly. Spectacular. Now, if these glands are just releasing their stuff into the interstitial fluid, do they require ducts?
No.
No. So these are actually ductless glands. I don't actually know if that's a word, but I'm going to use it as a word because it amuses me to do so. And of course, while we gave lots of examples of what exocrine glands secrete, what's the general term for the substances that endocrine glands secrete?
Hormones.
Exactly. All right, hormones. So we are going to worry about hormones. All right, excellent, excellent, excellent. Now, remember, these hormones are a specialized chemical signal used for communication. Of course, there was one other organ system that we talked about in this class that was specialized for communication. And what was that one?
Nervous system.
Nervous system, absolutely. So our endocrine system uses hormones, which are basically a chemical signal to communicate. What does the nervous system use to communicate?
Action potential.
Yeah, action potential. Oops. Which is basically an electrical signal. So both of these are used for communication. Of course, what's the big difference between the two, or one of the big differences between the two?
The speed.
Speed, right? Probably the biggest difference between these two are their speeds, right? Both are used to communicate to the cells of the body, to coordinate their activity, but that electrical signal, like flipping the light switch in your room, is practically instantaneous, right? Fractions of a second, which is useful for rapid responses to changes in our environment, right? Whereas it takes a lot longer, as we learned in 430, for a cell to make a hormone, right? First, you have to decide you need to make a hormone. Then you have to find the right piece of DNA. Then you have to use a bunch of enzymes like RNA polymerase to make some RNA, to convert it to messenger RNA, to ship it outside of the nucleus to the rough endoplasmic reticulum, where a ribosome can find it, and translation can take place to make a sequence of amino acids, which then has to go to the Golgi apparatus, be cut, folded, modified, added to other pieces, bundled up into a vesicle till it can finally be released from the cell. Right? Think if you had to do all of those things when you put your hand on something hot. Those damaged cells had to make a hormone, send it to the brain, which then had to make a hormone to send it back to the hand, right? Your hand would be burnt off by the time that you got the signal to move it. So the endocrine system, these hormones, these chemical signals are a much slower system, but their power comes from those hormones. Many of these hormones are long-lasting chemical signals. And because they're long-lasting, they allow us to coordinate big, long, complicated processes over a big, long period of time, right? How many of you are the same size now as you were when you were born? Anyone? If so, I feel very sorry for your mom, right? No, we've all grown significantly since then, right? That ability to grow from that tiny little baby to the big, huge adult that you are. The conversion of the process from going from a girl type person to a woman type person, or a boy type person to a man type person. Those biological changes that take place, those are all things that take place over years, over months, over weeks, right? And it is those long-lasting chemical signals that are used to coordinate that type of activity. So both are important, both are involved in communication, but on very, very different scales of time. Again, it is an organ system, so we are going to have those organs, and most of those organs are going to be glands that are going to produce and release those hormones. And of course, the study of the endocrine system is the field of endocrinology. All right. So we kind of already hinted at this. Our hormones are chemical signals that allow us to respond to our environment. And like our nervous system, right, they combine to form our command center. If you think back to when we talked about the process of homeostasis, right? We have a disturbance, we have some receptor that perceives that disturbance, and then we send it to a command center. And that command center is where we process that information, make sense of that information, and then send a signal out to the effector. And if you remember, our command center is some combination of our nervous system and endocrine system. So when we're maintaining homeostasis in the body, whether it's metabolism, whether it's our biological clock, whether it's maintaining electrolyte levels in our blood, stress, temperature, all of these types of homeostatic processes are controlled by hormones. But as we also talked about, big, long-lasting physiological changes as well. Our growth, our maturation, and then also the propagation of our species are all controlled by hormones as well. When we typically think about a hormone, we've talked, as we just mentioned, it is typically a chemical signal, a long-lasting chemical signal, so a sturdy, long-lived molecule that is produced by one cell or one clusters of cells, right? Either a single cell or a gland, which is basically a cluster of cells. As we see, it releases it into the interstitial fluid. Without a duct. And from there, it can get into the blood. And as we talked about, when it gets into the blood, it can go everywhere. Now, if that goes everywhere, then that means potentially it could affect every single cell in our body. Right? I think we mentioned this back in the first section, but I don't remember if we did or not. But if you remember, we talked about it, someone came up with the number that like every living cell in your body is something like 30 microns or less away from a capillary, allowing it to get the oxygen, the nutrients, and the chemical signals that it needs. So that means potentially every single cell of the body could potentially be affected by a hormone. Does that mean that every hormone affects every cell of our body?
No.
No, and the reason for that is the cells that are affected, and in fact, the only cells affected are those that have the special receptor for that hormone. And these cells that have this specific special receptor for that hormone, we call target cells. So while there is the potential, right, if you don't have the special receptor, you are not a target cell and the hormone won't affect you.
Would it be a specific target cell for that specific hormone, or if a cell has a target cell, any hormone can?
Great
impact
question. No, each hormone has its own specific receptor, which is, again, if you think about it, is what we want. We don't want either a cell that is going to be affected by all hormones or no hormones, right? We want, and in fact, I think we've got a pretty picture here that shows this right here. All right. Notice here we have 3 cells and 2 hormones. Notice the yellow cell here just has receptors for hormone A. So you could dump a whole bucket of hormone B on this yellow cell, and will it have any effect? No.
No.
Right? Conversely, green cell, we could dump a whole bucket of hormone A on it, and it's not going to have any effect because it only has the receptor for hormone B. But notice the blue one in the middle has receptors for both. And so it can be affected by both. In fact, in what we will see is the, let's say it this way. The actual effect of a hormone, we're going to talk about all the effects of the hormones. We're going to talk about that a lot for this next week. Really, the effect of the hormone is actually caused by the combination of the hormone binding to the receptor. And of course, as we know, anatomists then are going to have to give it a fancy name. And in fact, that's what they call it. They call it the hormone receptor complex. So it's produced by so this complex of the hormone and the receptor binding together is actually what causes the change in the cell. So if the cell doesn't have the receptor, it is not going to be affected by the hormone. Now, of course, it can't be quite this simple, right? Is like this yellow cell here. Was this yellow cell born with 5 receptors for hormone A on it, and it will always have 5 receptors for hormone A for its entire life? Let's think of it this way. These receptors are proteins on the plasma membrane. Oops, why did that not write? As we know, are the proteins on the plasma membrane of a cell static and unchanging?
No, it's
No.
dynamic.
It's dynamic. We constantly, remember, have the ability to make new vesicles that may have a particular protein on the plasma membrane that can come to the surface, and when it binds and becomes a part of the surface, that new protein is expressed on the surface. And by the same token, we've seen how proteins can be endocytosed, brought back into the cell. So their expression, the expression of these receptors are dynamic. They can be upregulated and they could be downregulated. Let's think about this. Here I have a cell, and this cell has, I know these look a lot like antibodies, but I'm just doing this for simplicity. These are not antibodies. These are our receptors. And actually, Let's avoid confusion. There's a receptor for a hormone. There's a receptor for a hormone. There's a receptor for a hormone. This cell happens to have 3 on them. Now, if I tripled this and now suddenly had 9 on the surface, if I just took a little bit of the hormone and sprinkled it onto this cell, you'd be much more likely to have an effect with 9 receptors versus 3. However, the opposite is true as well. If I were to go and remove all of the receptors from a cell except for 1, and then I dump a whole bucket of hormone on this cell, even though it's a whole bucket of hormone, is it going to have a big effect with just 1 receptor? Probably not.
No.
And let's take the opposite. If I remove all the receptors and I dump a bucket on the hormone on it, what would happen then?
Nothing.
Nothing. Notice that process of down regulation is a process we call habituation. Right? Again, you may not have thought of it in these terms as far as hormones and cells, but you're all poignantly aware of this, especially those of you who are parents, because when that child is a baby and they make the tiniest little whimper, right, that smallest little signal, and you respond to it, you're rushing into the room to see if your baby is okay. By the time they're 2, they could be sitting next to you screaming their bloody head off, but if you're on the phone, you're not paying any attention to them. You've completely habituated them. It's white noise, not even paying any attention at all. And the same is true with upregulation. With upregulation, we sensitize making it more sensitive. And again, if you think about it, we have the ability to make proteins. It may be possible to put a new protein on the surface of this cell that is a new receptor that it's never had before. And now suddenly this cell can react to a hormone that it was never able to react to before. So the take-home message for this is the presence of receptors that are necessary for that hormonal effect is the dynamic process, constantly being upregulated and downregulated. More receptors and less receptors will change the effect of the hormone. Now, the other thing I believe we've hinted at, because we talked about cytokines, is normally when we think of hormones, we think of them in terms of circulating hormones. Everything we've talked about so far in this section has been those circulating hormones, where again, hormone is made by one cell or a gland, gets into the blood, and goes everywhere. But if you remember in our immune response, we talked about cytokines, for instance. Cytokines are a type of local hormones. Local hormones are typically less stable. And because they're less stable, they're less long lasting. These aren't the kind of things that are going to be very useful to stick in the blood and try to send everywhere. Instead, as their name indicates, these are primarily used for the cells that are surrounding it. Right? For the local cells in the local environment, like in the bone marrow. This is how we trigger the production of a specific type of white blood cell. And in fact, if you think about it, when we talked about that helper T cell, what did we say the helper T cell, the first thing the helper T cell stimulates is? When those helper T cells release their cytokines, what did we say the first thing they stimulate was?
A rush of leukocytes, white blood cells.
and not just any white blood cells, but specifically the production of more helper T cells. Remember, the first thing we said the helper T cell does is stimulate itself, right? That ability to stimulate itself is what we call an autocrine. Paracrines would be, for instance, like what a helper T cell does to a B cell, where it is releasing a chemical signal and affecting a target that is nearby, right next to it. So these local ones have local effects. They're not going to be circulating. They're not going to go out into the blood and go everywhere. But they can have local effects and local cues in their local environment.
Can you repeat the paracrine one more time? What happens?
Paracrine is just the surrounding cells. So
Thank
it's
you.
just affecting the surrounding cells. All righty. So as I mentioned, we have to have that hormone receptor form a hormone receptor complex to have an effect. And many of these hormone receptor complexes form on the plasma membrane. What was the key word in that sentence?
Most.
Right, most or many, because that means not all. Some actually will enter the cell first and will actually bind in the cytoplasm. So they will either bind on the plasma membrane or inside the cell where they bind in the cytoplasm. And when that hormone receptor complex activates the cell, you will get one of several possible effects. It will either cause the making of new proteins. Of course, if you're making new proteins, remember, as we talked about, we can express those proteins on their surface. Putting a new channel or a new pump or a new receptor on its surface, which would change the permeability of the membrane. We can either increase or decrease metabolism, change the number of chemical reactions taking place. And of course, we're going to do that by turning enzymes on or off, right? Because as we turn enzymes on, the number of chemical reactions is going to go up, metabolism goes up. If we turn enzymes off, the number of chemical reactions go down, the metabolism goes down. And one of the things that makes these hormones so challenging, makes it so hard for us to both study them, right, as mature scientists and then also as us learning about this, is that each target can respond differently to a hormone. Let's talk about a really simple example. A hormone where most people are familiar with, insulin. I think we talked about this in the last class. What did we say insulin does? So if only we'd done this.
Glucose levels in blood.
Yeah, it lowers glucose levels in blood by telling cells to take the glucose out of the blood plasma and bring them inside. However, once that glucose is inside of the cell, does every cell treat that glucose the same way?
No.
No. So for instance, a liver cell will take that glucose and will convert it into glycogen, storing it that way. However, an adipocyte is going to take that glucose, bring it in, and it is going to stimulate the production of triglycerides, storing that excess energy as fat. So exact same hormone, but on different cells, it's going to have different effects.
Professor, did you say that upregulation is referred to as sensitized and downregulation is habitual?
Habituate.
Habituate, okay.
Yep. So this is part of the challenge. And good news is it gets even more complicated than that. So, as I mentioned, a hormone is going to have different interactions with our target cell. And again, this is going to be influenced by the hormone and the receptors concentrations. However, as I mentioned, one of the challenges is that hormones affect cells different ways, and hormones can interact with other hormones. Let's take a look at this. Let's use the whiteboard and talk about this. Again, this is a concept, a very important concept that we're going to talk about called hormonal interactions. Hormonal interactions is basically having to do with how one hormone can modify the effect of a second hormone on the target cell. All right, let's start easy. Here's my cell. Here's my target cell. And here's hormone A. And we'll make hormone A a circle. That, of course, means that my target cell, if it is a target cell for hormone A, must have a receptor that hormone A can bind to. All right. Now, when hormone A binds to the target cell, let's say that it doubles its metabolism. So, of course, when hormone A is by itself, what is the effect on my target cell?
Activates.
Yep, it's going to activate it. And what's the effect on the target cell going to be?
Excitatory.
Yeah. How excitatory? How much is it going to increase the metabolism?
Twice.
Yeah, it's going to double it. There you go. Wasn't a trick question. A by itself doubles the metabolism. So here you are in a lab. Your lab instructor, your primary investigator says, hey, I've got this cell and I've got these two hormones, and I want you to tell me how they affect them. So you have this target cell, you give it hormone A, and you see that it doubles the metabolism. Excellent. Check it off. End of story. Right? Then they ask you to come back and do the same thing. Great, you did such a fantastic job. I now want you to do the exact same thing with hormone B. Because this cell also has a receptor for hormone B. And hormone B just happens to be a blue square. And so on the first cell, you put hormone B on it. And it doubles the metabolism. Fun. On the second cell, nothing happens. On this one here, it decreases the metabolism by 1/3. Excellent. So now you're done. Except you're not, because it turns out that while A has an effect by itself, while B has an effect by itself, there is the possibility that A and B can interact with each other. So what happens if we use A and B together?
Is that the synergistic effect?
Well, we're getting there. Absolutely. You are correct. This first one is indeed a synergistic effect. And what is that synergistic effect?
Metabolism will increase 4 times.
So you've got the right idea. Absolutely. What is going to happen here with the synergistic effect is that we are going to get, notice, 2 positives working together. Oops, hold on. However, in this case, instead of it just being 4 times together, it's now 10 times the metabolism. Notice the key with a synergistic effect is it is not just additive. If you think about it, if it was just 4 times, then really they wouldn't be doing anything to each other. What we're getting here is an enhancement. It's not just adding them together. We're getting an enhancement. We're getting a much bigger effect than either of them by themselves. All right, that is a synergistic effect. Notice on this cell here, we figured, well, B does nothing. We can just throw B away. We don't have to worry about it anymore. But something magical happens when we do put A and B together. In this case, we put A and B together, lo and behold, we get a 10-fold increase in metabolism. What the heck happened here? Is this also a synergistic effect?
Did hormone B enhance the effect of hormone A?
Exactly. Notice the key that happened here is B has no effect by itself. but it enhances the effect of the first hormone. And what kind of an effect would we call that?
Permissive effect.
Exactly. And this one's probably the most obvious. What's happening here? What's going to happen down here at the bottom?
Two hormones are going to work against each other.
Yeah. Notice what happens here is we have two hormones that are working in opposite directions. And so what we get is less of an effect of either by themselves. Right? We could have done this simply. If A doubles the metabolism and B halves the metabolism, what happens when you put them both together?
Nothing.
Yeah, they would cancel out and there would be no effect. Notice here, they're not going to fully cancel out. So as a result of this, we may get a small decrease in metabolism, but we've got less an effect of A by itself, less of an effect of B by themselves. So it decreases the effect. They're working against each other. And what do we call that? What's that fancy word we use for when these things are working against each other? There we go. Samuel's got it. It's an antagonistic effect. Oops. It's an antagonistic effect. So there's three possible ways a hormone can interact with another hormone. If they both go in the same direction, it's synergistic. If they go in opposite directions, it's antagonistic. But we also have this interesting third situation where we sprinkle a hormone and it doesn't appear to have any effect by itself. Right? This is one of the problems with this field. Think about you're studying a hormone for the first time and you sprinkle it on a little bit of brain tissue and nothing happens. And then you sprinkle it on a little bit of liver tissue and nothing happens. And you sprinkle it on a little bit of an eyeball tissue and nothing happens. And so you toss it away, figuring you don't have to worry about that anymore. But then you realize that while it may not have a direct effect itself, it could change the effect of some other hormone. So now you have to take your hormone and sprinkle it with hormone A and sprinkle it with hormone B and sprinkle it with hormone C. Or maybe it takes 3 hormones to get the interaction, right? It's not pretty, it's not simple, it's not easy. With this permissive effect, the hormone B by itself does nothing. But it does change the effect of another hormone. Right? So no effect by itself, but it does change the effect of another hormone. So 3 possible interactions. All right. So again, we come back to the pretty pictures and the words. Or synergistic, 2 hormones, same effect, work together to enhance, right? They have the same effect alone and enhance together, right? That is our synergistic effect. Permissive, second hormone, no effect alone. But it enhances the first. And lastly, antagonistic. 2 hormones with opposite effects are basically going to cancel each other out. And they reduce the effects of each other by themselves. All right. Questions on this? This doesn't make sense. It only gets worse from here. So if you have any questions, ask them now. All right. That is hormone interactions. But the other thing we have to worry about is hormone regulation. Now, is it important to maintain an appropriate amount of hormone in our blood?
Absolutely.
Absolutely, right? Your insulin levels, we want to stay fairly stable, right? So how are we primarily going to do that? What is the primary way, do you think, that we maintain appropriate hormone levels in the blood?
Aren't there chemical signals that are sent out to get rid of insulin in the blood?
True. And what would you monitor to be able to do something like that?
The
How would
amount,
you know if there was too much insulin in the blood?
well, you would have receptors
Receptors for what?
for insulin.
Exactly, right? If your body temperature goes too high, you have receptors that then cause you to sweat to bring it back down, right? What did we call that type of a process?
Negative feedback.
Exactly. The primary way we maintain levels of most of the hormones in the body is by using negative feedback. You're absolutely right. We have sensors for insulin, and if it goes too high, we stop producing it, we stop releasing it, we get rid of it in our blood, we get rid of it in our liver, and it comes back down. If it drops too low, we can make more, we can release more, we can bring it back up. So the primary way we are going to control hormone levels in our blood is negative feedback, because we want that stable amount of insulin. Except when we don't. When that little kid goes to the birthday party and eats the 5 pieces of cake and the 3 scoops of ice cream and gets the goodie bag full of candy that they eat in the car on the way home. What do we want to do to that kid's insulin levels after that?
Increase?
We want to increase them, right? So notice, while we primarily want to use negative feedback to maintain normal levels, there are going to be times when we want to change them, like after that birthday party. Right? There are times we need to modify the levels. And if we are going to modify the levels, there's basically one of 3 regulatory processes that we can use to adjust hormone levels as needed. Let's go back to our whiteboard and use the exact example we've been talking about. Again, now we are talking about hormonal regulation. Again, we know primarily negative feedback, but there are 3 regulatory processes that can be used to modify the levels. And let's talk about the one we were just talking about. For example, insulin. What produces insulin?
The pancreas.
Excellent. And when that kid went to the birthday party and ate the 17 pieces of cake and the 14 scoops of ice cream and the 3 bags of candy, what does the body do in response to that?
Produce more insulin.
Produces a lot more insulin. And what is the trigger? What is the regulatory process? How does that change?
The sugar level in the blood increases.
Exactly. We are able to measure the glucose levels in the blood. This type of regulation, where we are regulating the chemical, let's write it up here. Where it's regulated by, actually, I'll write it down here and then I'll adjust everything. By the amount of the chemical, that is present in either the blood or the lymph or the interstitial fluid, so that it's in basically the fluids of the body. And what was that fancy term we had for the fluids of the body?
Humor.
Humors. And that's actually what we call this. We call this humoral regulation. Humoral regulation is where we, let's say it this way, change the amount of a hormone based on the amount of a chemical that is in the fluids of our body. And the perfect example of that is the insulin, right? And here's the problem with that insulin. That kid went to the birthday party, had the 3 pieces of cake, the 2 scoops of ice cream, and the big bag of candy. And what happens to their blood glucose levels?
They go up
Just a little bit?
probably more than.
Yeah, no, they get a huge spike from eating all that sugar at once. And so when your body sees a huge spike of glucose, does it release just a little bit of insulin? No, it releases a lot of insulin. And remember, as we mentioned, insulin is a long-lasting chemical signal. So while that insulin is in the blood, it's going to stay there for a long time. And so while it's going to be able to do a good job of bringing the glucose levels of that kid back down to normal, is all that insulin simply going to immediately go away as soon as the blood levels reach normal?
No.
No. So what continues to happen to the blood glucose levels?
Goes down.
Yeah, exactly. There you go. They continue to drop. And what typically happens to the kid after that big, huge sugar rush?
Crash.
That sugar crash. Exactly. That sugar crash is caused by that decrease in glucose levels from that massive amount of insulin that was released into their blood. Excellent. Let's think of another example we are comfortable, should be somewhat familiar with, right? Again, you've got a group presentation in 1 week, you've got an exam in 9 days. So most nights, late at night, 1 o'clock in the morning, you should be sitting at the kitchen table studying anatomy and physiology. Because that's when everybody's asleep and the house is quiet and you can be productive. And while you're sitting there and the entire house is quiet and you're slowly studying at the kitchen table, there's this huge, massive bang outside. You don't know what's going off, right? Are you being invaded, right? Gunshots going off. What the heck is going on out there? And then you very quickly realize that it's just your neighbor's car backfiring. You know that you are perfectly okay, but does your heart rate instantly go back down to resting rate?
No,
No,
No.
your heart rate stays elevated for another 20, 30 minutes. Why?
because it's a long-lasting chemical signal.
What is the long-lasting chemical signal?
Adrenaline,
Adrenaline.
Adrenaline, absolutely. Or what's the other name for adrenaline?
epinephrine,
Epinephrine. And again, you may use adrenaline or epinephrine. I'm fine with either of that. Excellent. We have epinephrine. Epinephrine is produced by what?
adrenal glands,
Excellent. It's produced by the adrenal gland. Excellent. And what controls the release of epinephrine from the adrenal gland?
The sympathetic nervous system.
Your nervous system perceived a stressful situation, a potential risk to your health, and it sent an electrical signal, an action potential to your adrenal gland, causing it to release that adrenaline into your blood. And not surprisingly, that type of regulation is neural regulation. Neuroregulation is where you get a direct input from the nervous system, modifies the release of the hormone. And epinephrine is a good example of that. So now we've got humoral regulation. We have neural regulation. What is the third way we can regulate the amount of a hormone that is in our blood?
By other hormones.
Yeah. Our third type of regulation is hormonal regulation. This is where one hormone modifies the amount of a second hormone in our blood. Can anyone give me an example of something that might do that? I know we haven't talked about hormones yet, so it's not completely fair, but anyone peek ahead at the list? Let me give you a hint. What do you think thyroid stimulating hormone does?
Stimulates the thyroid to release hormones into the body.
It's hormones. And for right now, we'll get away with T3 and T4. We'll say what those are as well. Absolutely. So notice how much of the thyroid hormones are in your blood can be modified by thyroid stimulating hormone. Anybody know where thyroid stimulating hormone is made?
In the thyroid?
No,
Or the
not a bad
thymus?
guess. No. It's the anterior pituitary. Anterior pituitary is called the master endocrine gland. It produces over a dozen hormones, and many of them, like thyroid stimulating hormone, tell the other endocrine glands what to do. So we talked about the three hormonal interactions: synergistic, permissive, and antagonistic. We now have three hormonal regulations: humoral, neural, and hormone. And if you notice, On your list of hormones, there is a column regulated by. So the good news is every single answer in that column is going to be one of these three words: humoral, neural, or hormonal. So if I, for instance, asked you how insulin was regulated, what would the correct answer for that be?
Humoral.
And you would get partial credit for that on the exam. What else do you think you need?
Regulation.
Nope.
Is it both humoral and hormonal?
Humor, remember, blood is a humor in the body. So blood falls into humoral. Now, you got the right idea, Amanda, before. You absolutely positively need to say humoral, neural, or hormonal, but I also want what specific chemical in the blood is it that we're using for regulation. So it's humoral via what?
Glucose.
Glucose. If instead I asked you how epinephrine was regulated, what would your answer to that be?
Neural via sympathetic.
There you go. And if I asked you how T3 was regulated, what would your answer there be?
Hormonal via thyroid stimulating hormone.
Exactly. So that's what I'm looking for. One of these three regulatory processes, but you have to tell me what the chemical is, or tell me what branch of the nervous system it is, or tell me what hormone it is that is controlling it. I want both parts of that. Does that make sense?
Professor?
I'm sorry?
Sorry. Is there any example for neuroregulation or?
Yeah, the adrenal gland is controlled by the nervous system. There are other examples as well that we will run into them.
Because I think the main one we always talk is the adrenaline.
Yep.
But I don't know, I've never heard of that.
There's two more we'll talk about at least. Two more. So it's not as common. It's more humoral and hormonal, but the neural is important. Actually, that's not true. There's three. Three more, four total. I think there may be more. We will go through them, I promise. Let's talk about some examples from your textbook, because your textbook does a good job of talking about this as well. So again, we cannot forget that the primary way most hormones are regulated is by negative feedback. All right. However, there are these important other types. Hormonal control. Notice in this case, and we'll talk about this mechanism in detail, our hypothalamus happens to produce a hormone, for instance, that is called thyroid stimulating. Oops, hold on. Make this way smaller. Releasing hormone. And guess what the thyroid stimulating hormone releasing hormone does?
Stimulates the hypothalamus to release the thyroid hormone.
It stimulates the pituitary gland to release thyroid stimulating hormone. And what did we say thyroid stimulating hormone did?
Tells the thyroid to produce T3 and T4.
Exactly. So notice here we actually have two examples of hormonal control in a row. Hypothalamus telling the pituitary what to do with a hormone, and then the anterior pituitary telling the thyroid what to do with a hormone. I did warn you that this got a little complicated, right? It's not, so basically each of these has their own effect. Notice in particular, thyroid stimulating hormone, both hormonally controls the thyroid gland, but is also hormonally controlled itself. And we'll go through all of these, like I said, together. We just need to understand these. Humoral, we talked about insulin. Notice here our parathyroid gland produces parathyroid hormone in response to changes in calcium levels. If you remember from when we talked about the bones in 430. So as calcium levels change, we can release more or less parathyroid hormone in response to those changes in calcium levels. And as we also talked about, our adrenal gland, specifically the medulla of the adrenal gland, is stimulated directly by the sympathetic nervous system. Notice conveniently enough, they've actually given us that preganglionic neuron whose cell body is located in a lateral gray horn that will go out and stimulate the medulla to release that epinephrine and also norepinephrine into our blood. So 3 main types of modifying hormone levels, 3 main regulatory processes: humoral, hormonal, and neural. And every hormone on our list will have one of these. All right. Questions on that? All right, excellent. This is a good stopping point for our first break. Let's go ahead and we'll just switch this. This is a good place for our first break. So let's go ahead and take our first break. We'll take a 15-minute break, a full 15-minute break, because again, today is going to be a long day. So that's going to be 1:22 that we will restart. And I will start the recording at that point. All right, any questions before we take our first break? All right, I will see you guys in 15 minutes. All right. So what we need to talk about next is that all hormones are not built the same. There are 2 main classes of hormones, and you will be responsible for this information. So for each of your hormones, you do need to know which of these 2 classes it is going to be. And those two classes are amino acid based and lipid based. Now, again, these are major classes. So not surprisingly, there are lots of specific types and different types that I won't hold you responsible for. But for every hormone on your list, you should know which ones are amino acid based and which ones are lipid based. This is important because the 2 different classes of hormones have different effects on the cells. One, they're going to bind in a different location. Some are amino acid based, bind on the plasma membrane, whereas lipid based bind inside, which if you think about it makes sense. Lipid based hormones, your plasma membrane is primarily lipids, phospholipids with that phospholipid core. And we know lipids like lipids, so it can pass through without any problem that way. But they're also going to differ on how that hormone receptor complex is going to affect the cell as well, right? Is it going to increase or decrease metabolism, or is it going to lead to some type of gene expression or something along those lines? So let's talk about these. Now, as I mentioned, there are different types of amino acid based hormones. I'm going to describe some of the different types. You don't need to know, for instance, that triiodothyronine, which after you've written that out once, and again, that is definitely something you'll have to spell in the exam, you can abbreviate it T3. You don't need to know that it, for instance, is amino acid derived. You just need to know that it's amino acid based. And basically what this means is it is based off of an amino acid. We have those 20 amino acids with functional groups. And basically what we do is restructure them functional groups. And not surprisingly, triiodothyronine has 3 iodines added to it, which is where it gets its name. So thyroxine, triiodothyronine, melatonin, epinephrine, norepinephrine, all of these are examples of small little amino acid-based hormones. However, we can put many amino acids together to form a peptide strand, and these form peptide hormones, right? Again, you don't necessarily need to know that oxytocin is a peptide hormone, just to know that it is amino acid based. One of the interesting things about these peptide hormones is they're often synthesized in an inactive form, and once released, they're then modified into their active shape. Thyroid stimulating hormone, luteinizing hormone, anti-diuretic hormone, human growth hormone, all of these are examples of those. There are also different types of lipid-based. The most common lipid-based hormone is a cholesterol-based hormone, and these are what we commonly refer to as steroid hormones. Notice this here is your friend and mine, cholesterol. We talked about him once before. He's got those 4 carbon rings. And as we talked about, he was located within the phospholipid tails of our plasma membrane, giving a little bit more structure and integrity to our plasma membrane. But again, by changing the functional groups, by changing these side chains, we can form different types of steroid hormones. Notice here, we have 2 incredibly similar cholesterol-based steroid hormones, just some minor differences in their side chains. So that must mean they must be very similar in their function as well, correct?
No.
No, and in fact, these 2 hormones right here, one of them is an estrogen and the other is testosterone. Right? They don't get much differenter than that. Right? One turns girl type people into women type people, one turns boy type people into men type people. So notice even subtle changes to these side chains can have dramatic influences and effects on what they can do to the body. The good thing about our steroid-based, our lipid-based hormones is they're only produced in a couple locations. So remember, I mentioned that you need to know which ones are amino acid based and which ones are lipid based. But here's the good news. The lipid based hormones are produced by the reproductive organs, the ovaries and the testes, the cortex of the adrenal gland, and the kidneys. So all the hormones we're going to talk about from those 3 locations are going to be the steroid based hormones, lipid based hormones. And every other organ we're going to talk about produces amino acid based. So it isn't too scary after all. Now, there is one issue with being a lipid-based hormone. If we have a lipid-based hormone, do we want to just take that hormone and dump it into the blood and hope for the best? No, because things are transported in the blood by the plasma, by water. And do lipids and water like each other?
No.
No. So typically, there are going to be some type of transport proteins that are going to bind to these hormones as they're transported throughout the body. So we have to take these water insoluble steroid hormones, connect them to a polar protein, and this allows them to be dissolved into the blood plasma and transmitted throughout the body. So our steroid hormones need some type of transportation proteins to get where they need to go. Now, remember, I also talked about how some local hormones are less structurally sound, and so they're only able to have local effects, and they're not circulating hormones. Many of those fall into the lipid-based types. Notice here we see an example of one of these, a class of hormones called echocyanoids. Notice in this case, they are formed by just a single carbon ring and a few fatty acid tails coming off of this. right? This is much, much less structurally sound than that 4-carbon ring cholesterol. So it's not surprising that these are not going to be as sturdy. These are not going to be the kind of things that are easily going to be transported into the blood and sent throughout the body. So these echocyanoids are primarily the paracrines, prostaglandins, which we'll talk about. Many of our cytokines are echocyanoids and things along those lines that allow for that local communication. And that's because they are so much less structurally sound. All righty. Now, let's talk about the hormones. As I mentioned, let's do it this way. Actually, you know what? I lied. Let's go back here. When we are talking about our amino acid based and our lipid based, we said there are some key differences. For the lipid base, what were the 3 organs where we said they were produced?
The sexual reproductive, the adrenal cortex, and
Kidneys.
Excellent.
the kidney.
So we'll cheat, and for amino acids, we'll just say produced everywhere else. But there are a couple other big differences that we talked about as well. The first is going to be where it binds to the receptor. Our lipid-based, remember, because they're lipid soluble, they are going to bind to the receptor inside the cell. This can actually be in the cytoplasm, or in some cases, it actually can be inside the nucleus. Amino acid bases are not lipid soluble. So because of this, they bind to the receptor on the outside, on the plasma membrane. Now, let's think about this for a second. Our lipid base, they're inside the cell. Since they're inside the cell, they can actually directly affect the function of the cell. If you're outside the cell, can you directly affect what's going on the inside?
No,
No.
it's to affect the receptors first.
Right. However, right, when the Amazon delivery guy comes, which he does at least 3 or 4 times a day, right? Even if he doesn't ring the doorbell, even if he doesn't knock. Now, especially if he rings the doorbell and especially if he knocks, but even if he doesn't ring the doorbell and doesn't knock, how do I know he's there?
Because you get a text telling you that your package
True.
has been.
That is possibility. Packages at the door. I could have a ring. Maybe I should mention the fact that I have 2 dogs. How do I know when the delivery guy shows up, even if he doesn't ring the doorbell or it doesn't knock?
They start barking.
Dogs go crazy on the inside. And my dog in particular will run around the house, screaming and yelling and barking and hollering, knocking things over, causing quite the mess. So while that delivery guy cannot directly affect what's inside the house, does he, through a second messenger, in this case, my dog, make me aware that he's there? Yeah, and that's exactly
Yes.
what we call this. In this case, we don't directly affect the inside of the cell, but what it does do is it influences what we call a second messenger. And that second messenger is then going to influence the direct change. So he knocks on the outside door, rings the doorbell, and the hormone does, and then that second messenger, that dog, goes crazy on the inside. The last big way that they are different is that amino acid-based hormones change the metabolism of the cell. Now, let's think about this for a second. If you change the metabolism of the cell, how many ways are there to do that?
3.
Okay, what are the 3?
Increase, decrease, stop.
Well, you could argue that stop is just a dramatic decrease, right? Starting would just be a dramatic increase. So let's just go with 2. Excellent. So amino acid-based hormones are going to either increase or decrease metabolism. And remember, as we mentioned, what that means is that it is either going to turn enzymes on or off. Notice our lipid-based, not only do they bind inside the cell in the cytoplasm, but notice I also said they could actually make it all the way to the nucleus before they bind. And that is because what our lipid-based hormones do is they basically trigger gene expression. And someone again remind me what genes express? What does that mean to express a gene?
Is it DNA?
Yep, it is part of the DNA. And the
Synthesis.
gene is that meaningful part of the DNA that gives us the instructions for doing what?
Synthesize proteins.
Exactly.
That gene.
Triggers gene expressions, which is basically causes us to make proteins. Absolutely. So when we talk about our 2 main classes, amino acid based and lipid based, this is the big differences between them. And do you think the pathways by which they do these 3 things, either increase the metabolism, decrease the metabolism, or make proteins, are going to be important?
Yes.
Yes. In fact, so important that I would be shocked if one of them wasn't an essay question on the exam. So let's take a look at this process. As we talked about, amino acid-based hormones are going to change the metabolism of the cell. They're going to either turn enzymes on or turn enzymes off. And again, remember, that's what metabolism means. Metabolism is basically the number of chemical reactions. That's essentially what we mean when we say metabolism. It's the number of chemical reactions that are taking place inside of a cell. So as we know, enzymes speed up chemical reactions, make them easier to occur. So if we activate a bunch of enzymes, we get more chemical reactions. If we shut down a bunch of enzymes, we decrease the number of chemical reactions. So we are going to change the metabolism, turn these switches on and off. Because they're amino acid based, they're not able to enter the cell. So they cannot directly influence the inside. So instead, they have to require a second messenger and a series of specialized proteins called G proteins. Now, many things can actually be second messengers inside of a cell. Here, again, our delivery man binds to our receptor, rings the doorbell, and then the dog on the inside goes crazy. Notice in this type of format, our receptor is an integral membrane protein. What does that mean?
Protein is integrated inside the membrane.
Yep, and in particular, it's a transmembrane protein. What does that mean?
It goes through the plasma membrane.
It goes through both layers. Remember, integral means that it is attached to the plasma membrane. You could have an integral membrane protein that was just embedded in one layer of the phospholipid. But to be a receptor, you have to go all the way through. You have to be a transmembrane protein. Notice this G protein is a peripheral membrane protein. What does that mean? Anyone remember? I know it's been a long time since you thought about these things from 4:30.
It is only expressed on one side of the membrane, doesn't actually go through it.
So I like the second part of what you said there. Absolutely. A peripheral membrane protein is not directly connected. It is associated with the plasma membrane, but not directly connected. And the advantage in this case of it not being directly connected is it actually is, when activated, becomes a motor. Remind me again what a motor is?
It moves something.
Yeah, and more particularly what it does is it uses a chemical energy to produce a mechanical energy. So it converts a chemical energy into mechanical energy. So you've absolutely got the right idea. Basically, when activated, it is going to move along the plasma membrane of the cell. Now, there are several things, half dozen things that can be second messengers in the cell. Calcium is an example. Cyclic GMP is an example. However, the most common and the one we're going to use for both of our examples is cyclic AMP. AMP stands for what again?
Adenosine monophosphate.
And adenosine monophosphate, absolutely. Basically, it is a adenine nitrogen base, a ribose sugar, and a single phosphate. We don't have to worry about what cyclic means. From a chemical standpoint, we don't have to worry about that. But it's a modified energy molecule, like a DP and a TP. All right. So it's a special modified energy molecule, cyclic AMP. And so for both our excitatory and our inhibitory processes, cyclic AMP will be our second messenger. All right. What we're going to do first is, again, we have three processes we have to learn. So we are going to focus on the first one being excitation, increase in the activity. Let's go to the whiteboard for this. So, process one. So using an amino acid based hormone, we are going to figure out how to increase the metabolism. And again, remember, this is an excitatory process. We're increasing the process. Now, obviously, the first thing we need is our plasma membrane. We know it's a phospholipid bilayer, but I'm just going to draw it as a line to make my life easier. We already know we have to have a transmembrane protein. That is our receptor. And as we also mentioned, and I'll cheat and I'll use a circle for this, there is going to be a peripheral membrane protein associated with this called a G protein. So this is my receptor. Right? This is the outside of the cell. This is the inside of the cell. And this is our G protein. And at this starting point, it is inactive. And as someone mentioned, this is a peripheral membrane protein on the inside of the cell. All right. So this process starts with our hormone binding to the receptor. So of course we have an amino acid based hormone binding to the receptor on the outside of the cell. And of course, when these two things come together, what do we call them again?
Receptor hormone complex?
Yeah. So it forms the hormone receptor complex. Excellent. Now, in this case, when this hormone receptor complex forms, what it is going to do is it activates the G protein. And as we mentioned, this G protein, basically when active, is going to start to move. That active G protein, being a peripheral membrane protein, will continue to move along the surface of the membrane. Now, do you think it's just going to move along the plasma membrane forever?
No.
No, it's looking for something. And it turns out what it is looking for is for another peripheral membrane protein. Sorry, integral membrane protein. Integral membrane protein is, of course, embedded within the plasma membrane. And in particular, the specific integral membrane protein it is looking for is called adenylate cyclase. Now, with a name like adenylate cyclase ending with that ASE, what does that tell you?
It's an enzyme.
It's an enzyme. And at first it was inactive. But when our G protein finds it and binds to it, and when it binds to it, it activates. It activates the enzyme. So then the question becomes, what does this enzyme do? Well, it turns out what this enzyme does is it takes ATP molecules and it converts them into cyclic AMP. Remember, cyclic AMP is our second messenger. And if I'm converting ATP into cyclic AMP, what's happening to the amount of second messenger I have inside my cytoplasm?
increasing.
Yeah. So we get an increase in the amount of cyclic AMP. More second messenger. This second messenger, cyclic AMP, notice is the first molecule that is not actually bound to the plasma membrane. So it is able to freely move around inside the cell. And as it's moving around inside the cell, it is looking for a special protein called a kinase. And again, with a name like kinase, guess what it is?
Another enzyme.
An enzyme. And these kinases become active. And these kinases can then have a massive number of effects. They are going to turn on enzymes. And of course, if I turn on enzymes, what happens to my metabolism?
Increases.
It can actually open and close channels on the cell, and that can lead to changes in the permeability of the cell. And as we know, as we open and close channels and change the permeability, again, this can change the function of the cell. So we get more activity in the cell. And as a result of this, we are getting an increase in metabolism.
What was the first messenger, Professor?
Great question. You're right. We didn't mention that. What is the first messenger?
Is it the hormone, amino acid?
Yeah, exactly. The amino acid based hormone is the first messenger. Right? It can't enter the cell, so it needs a second messenger inside of the cell to affect its change. And notice one other important thing about this process. We describe this process as being a cascade effect. What does that mean again?
One event leads to another, leads to another.
Exactly. With amplification. What does that mean?
Each step gets bigger or has more.
You got the right idea. It doesn't necessarily have to be each step, but you absolutely have the right idea. Because let's think about it. And change the color just to emphasize this. How many hormones bound to my receptor?
One.
One. Which led to the activation of how many G proteins?
One.
Which led to the activation of how many adenylate cyclases?
One.
One. But that one adenylate cyclase, is it just going to convert one ATP into one cyclic AMP?
No.
No.
That would be several.
Exactly. And not just several, but probably many. And if we have many cyclic AMPs, we can affect many kinases, and we can affect many influences on the cell that are going to be able to give us a big, huge effect. So notice just the single binding of one hormone has the potential to have a major effect on the cell overall because of this cascade effect with amplification. I've done an amazing job of drawing this on the board, as I always do, but let's look at the pretty pictures from your textbook. Again, this one's actually not from your textbook. Your textbook does have a fun one though as well. We'll look at this one. This is a very simple one that I like. And it fits on the screen the right way, so that's why I use this one. Again, first possible pathway is that excitatory effect where we're increasing metabolism. Again, notice it starts with our hormone binding outside of the cell and activating the G protein inside the cell. Now, notice what this simple illustration doesn't show is that our G protein has to move down the plasma membrane until it finds the adenylate cyclase. The picture from your textbook, though, does a better job of showing this. But what this does do a nice job of showing is that that activated adenylate cyclase is going to convert ATP into our cyclic AMP, which again is the first molecule that's able to move around inside the cell, and that is why it is the second messenger. Notice the G protein isn't able to move around the cell. The adenylate cyclase isn't able to move around the cell. The cyclic AMP, like the dog running around the house, is the first one that can run around the house and affect the changes. Cyclic AMP activates our kinases. And those kinases, like we said, open ion channels, changing the permeability, changing the activity, activating enzymes, increase the metabolism. And we get that cascade effect with amplification. All right. Questions on that? All right, if this one makes sense, then let's talk. Oh, here's the bigger picture from your textbook. Clear all of that. Notice this one does a much better job. This is the one from your In Focus in your textbook, and it does a much better job of showing it. Again, hormone binds to that integral membrane protein, that transmembrane protein, the receptor, activates our G protein inside the cell, which migrates along the plasma membrane till it finds the adenylate cyclase, activating it, converting our ATP into cyclic AMP, which activates our kinases, which again stimulates enzymes, plasma membrane, open ion channels, all those things, all that activity of the cell.
And this is like a cycle too, like those other hormones kind of waiting in line, like once one's done, this keeps repeating itself, or?
Great question. No, and that's actually great feed into what we are talking about. Remember, as we said, hormone receptors are specific. So in this case, we have this hormone receptor that is specific just to this first hormone, hormone A, right? That hormone A that we talked about before. Hormone B then is going to bind to a different receptor, activating a different G protein. And that different G protein is going to go looking for a different integral membrane protein to activate. And that's how we're going to get a different effect. It's exactly how different hormones have different effects, because that's exactly what we're going to do next, our inhibitory effect. Now let's go back to the whiteboard. Plasma membrane. What's going on there? Plasma membrane. Again, this is the outside of the cell. Whoops, let's move this down a little bit. Inside of the cell. All right. Second pathway we care about, decreasing metabolism of a cell using an amino acid-based hormone, giving us that inhibitory effect. Notice again, this time we're going to need a different receptor for a different hormone. So let's go ahead and make this one a square. It is going to be bound to a different G protein. Again, there's some things that are the same. Our receptor is a transmembrane protein. Our G protein is a peripheral, a membrane protein. And again, at the start, our G protein is inactive. But as you guys mentioned, this is now a different receptor for a different hormone. So now hormone B, which again is our first messenger, is going to bind to our receptor. And of course, when it binds to the receptor, what does it form?
Hormone receptor complex.
Excellent, forms that hormone receptor complex. And when it forms that hormone receptor complex, it is going to activate the G protein. And when the G protein is activated, what does the G protein do?
Moves along the plasma membrane.
Moves along the plasma membrane. Not randomly, it's looking for something. In this case, it's looking for a particular integral membrane protein. But this one's different from the last one. In this case, it's binding to an integral membrane protein called phosphodiesterase. Now, the good news is once you've written out phosphodiesterase once, you can abbreviate it PDE. Again, when our active G protein binds to the phosphodiesterase, it activates the enzyme. However, what phosphodiesterase does is it takes cyclic AMP and actually converts it into its non-cyclic form. It converts it into just plain old AMP. In this case, what happens to the amount of second messenger? Remember, cyclic AMP is still our second messenger. This is the molecule that can move freely around the cell. But what's happening to the amount of our cyclic AMP now?
Going
Amount-
down.
The amount of cyclic AMP that is available in the cell decreases. So we have less of a second messenger. Now, with less of a second messenger, then that means fewer kinases can be activated. And if fewer kinases are activated, then that means fewer enzymes are turned on, fewer channels are opened, less activity in the cell. And what is the overall effect of all these changes?
A decrease in metabolism.
Exactly. The overall effect is a decrease in metabolism. And notice once again, we are getting a cascade with amplification. Because again, one hormone can bind and activate one G protein, which will then activate one phosphodiesterase. But is phosphodiesterase going to get rid of just one cyclic AMP?
No, it'll be
We're gonna
many.
lose many cyclic AMPs, which means far fewer kinases, far fewer enzymes. And again, one single hormone binding can have a huge, huge overall effect.
So
right. Yep, go
is
ahead.
this like a reaction to balance the cell after the first step, or these are just different hormones impact the cell differently?
It can be both. Different cells impact the cells differently. Now, is it possible that a cell producing insulin and releasing massive amounts of insulin, and then we want it to stop? So could we then send an inhibitory hormone to tell it to stop? Sure. Or something like that. Yes. I mean, it can be in response to, but it doesn't have to be in response to. So yes, these are completely independent processes.
Cool. Thank you.
Yep. However, if you think about it, if we had a cell that had both of these, then that would definitely be that antagonistic interaction that we talked about, right? Excellent. All right. We've done it here on the board. Let's look at the pretty pictures from your textbook. Again, this one's not your textbook. It's a simple one. But we have the same thing. Binding outside with the first messenger. Activating the G protein, which finds phosphodiesterase. Converting cyclic AMP into just AMP, giving us a decrease in the amount of cyclic AMP in the cell. So fewer kinases are activated, decrease in activity, and the overall metabolism decreases. And I think your book has a pretty picture for this one as well. Nope, I lied, it doesn't. Okay, so that's why I've got this simple one. But you get the idea. All right.
I have a question.
Yes.
How does the, like, what regulates what the G protein is looking for?
So, great question. The simple answer to that is the same way that we've been showing our receptors of having a specific binding site where only one hormone will bind to it. Our G protein at the end of it has a special key. And that special key is only going to fit inside of the lock of one door. So if this particular G protein here ran into an adenylate cyclase, nothing would happen. It doesn't have the right key to open the door. The only door this particular G protein can open is the door on phosphodiesterase. So even though we just use this generic term G protein, each G protein in this process is a unique protein so that it will be the same way that the receptor is a unique protein, so that it can only bind with one specific enzyme to activate it. It's a great question. Any others? All righty, excellent. With that, we are done with the two possible ways that an amino acid-based hormone can influence its target cell, but we can't forget about our lipid-soluble hormones, right? The process is going to be a bit different here. So let's go ahead and look at our third possible essay question. And that is the effect of a lipid-based hormone. And remember, as we mentioned, the fancy term we used for this was that it triggers gene expression. Which is a fancy way of saying what? Come on, what does gene expression mean? Yeah, make a protein. Tells the cell to make a new protein. This actually answers a question we had a long time ago in 430. Let's draw our cell. Let's draw our nucleus. What does our nucleus have inside of it again?
DNA.
DNA, absolutely. How is that DNA arranged in the nucleus? Is that all bound into these very precise books stuck on a bookshelf inside there? 3-ring binders?
Isn't it all kind of jumbled full together?
Yeah, it has some proteins that help it, but for the most part, it's a big, huge, loose jumble of material. And what did we call that big, loose jumble of material again?
Chromatin.
Yeah, there we go. Chromatin. Remember, chromatin is basically that loose DNA and proteins that are hanging around here inside of us. And someone remind me how many strands of chromatin I have inside of this nucleus?
2.
Just 2?
Oh, 30. I don't know. 30, 36. No,
36?
32.
32. 32? How many? Okay. What happens to
34?
our what happens to our chromatin when it is time for the cell to divide?
Duplicates. Replicates.
Okay, true, it duplicates, but that actually happened in interphase, right? In S phase. What happens in prophase when the cell is getting ready to divide? There we go. It condenses down into chromosomes. And someone remind me how many chromosomes you have?
23.
23 that you got from mom, but you also have,
23 from dad.
so you have 23 pairs. So how many total chromosomes are that?
40.
46, absolutely, right? So you have 46 loose strands of DNA and protein dangling around here. And if you remember back in 430, when we actually learned about transcription and translation, right? We learned that on these DNA are things called genes. And as we know, genes are the instructions for making a protein. And every single cell in your body, toenail cell, liver cell, eyeball cell, Do they have the same DNA or different DNA?
Same.
Same.
So does that mean toenail cells, liver cells, and eyeball cells make the same proteins? No, right? The instructions for making all the proteins are there, but each one is going to make different types of proteins. And do you remember how we said we were going to tell which proteins a cell should make? I'll give you a hint. What are we talking about now?
Hormones.
Hormones. Remember, we had another problem we talked about as well. Remember, as we mentioned, that only about 1.5% of our DNA is actually genes. 98.5% of our genes, of our DNA is stuff that they call junk DNA. For years and years and years, we didn't think it did anything. There's a whole actual new field of study called epigenetics that is looking at the potential implications of this quote unquote junk DNA and what kind of effects it could be having on our body. We don't care about any of that, but we still have this issue. As we talked about in 430, then, if I took a dart and threw it at my DNA, what are the chances that I will A, find a gene, let alone B, the precise gene I want that cell to make?
Very unlikely.
Very unlikely. Now, if you also remember back in 430, one of the things that we talked about is one of the ways we made this process easier is if, for instance, this strand here happens to have the particular piece of information I need. Remember, there's a special sequence of nucleotides called a promoter region, right? And then that tells us to start at that point. There was another special sequence of amino acids called a termination sequence that told us where to stop. And so our enzyme, remember that RNA polymerase would be able to bind to that promoter region, start making the copy to make the messenger RNA, and end at the end. However, every gene has a promoter region. Every gene has a termination sequence. So how does our ribosome, our RNA polymerase, know which one to bind to?
Was it supposed to be unfold on that region?
Well, but remember, when the cell's in, when the cell is in interphase, when it's making proteins, it is loose. We have the loose chromatin. So yes, we have a very few genes. Yes, they have these promoter regions, but how do we tell which precise one to make? Well, that's where the hormone comes in. These lipid-based hormones, these are the ones that are going to be responsible for telling the cell which specific protein it should make. Obviously, it starts with the protein coming in, pardon me, not the protein, the enzyme coming in, the hormone. So here's our hormone. Does this lipid-based hormone need a receptor on the plasma membrane?
No.
No. It's going to be able to pass right through the plasma membrane without any problems. Because it's lipid-based, it diffuses across via just simple diffusion. Doesn't need a receptor, doesn't need anything else. Once it is inside the cell, and I'm going to change the scale of it a little bit just to make it a little bit convenient for me, it is then going to bind to its receptor. So here is its receptor. It binds to this receptor. Again, this can happen in the cytoplasm or in the nucleus. And of course, when it binds to it, it forms our hormone receptor complex. This hormone receptor complex is then going to enter into the nucleus. Where, conveniently enough, there is this RNA polymerase. Oops. Now remember, the RNA polymerase can bind to any promoter region. So how do we tell it which one to bind to? Well, we use that hormone receptor complex. What's going to happen is that hormone receptor complex, and I'm just going to draw it as a red triangle now, a red rectangle, is going to bind to the DNA. So our hormone receptor complex, going to bind to the specific gene we want expressed. And the binding allows the RNA polymerase and all the other special proteins start to read the DNA, and using complementary base pair rules, start making a single strand of RNA nucleotides that matches that template strand. And what was the fancy term we had for that process?
Transcription.
There you go. Excellent. And from transcription, what do we get?
RNA.
Excellent. The end result is RNA, which is then modified into what?
Messenger RNA.
Excellent. What does our messenger RNA do?
Leave the nucleus, enter the cytoplasm.
Excellent. And when it leaves the nucleus and enters the cytoplasm, where does it go?
Endoplasmic reticulum, rough.
Excellent. And when it goes to the rough ER, what happens to it there?
Translation.
Excellent. And what is the end result of translation?
Protein with its primary structure.
Okay, I like that. I'll accept that. I was thinking more of just an amino acid strand, or a polypeptide is probably, but I would totally accept that. The primary structure of a protein. So that polypeptide, that single strand of amino acids, where does it go?
Golgi apparatus.
Excellent. And what happens at the Golgi complex or the Golgi apparatus?
Modification.
Yeah, it's cut, it's folded, it's added to other subunits. And what's the end result?
Protein that has a function.
Yeah, a functional protein. And then that functional protein, as we know, can either be expressed on the plasma membrane, or it can be released from the cell as a secretion. All right. Any of that vaguely familiar from 430? So notice in this fashion, we finally learn how we tell the cell what gene to read, what protein to make, and when to make it. Because we always have that RNA polymerase and we have all those promoter regions, but it's not till our, and let's do a better job with this. It's not till our hormone receptor complex comes in and binds to the DNA that our RNA polymerase can come up and start reading that particular strand, making that messenger RNA, sending it to the rough endoplasmic reticulum to make our polypeptide, to send to the Golgi apparatus, to make a functional protein, to then either put a new protein on the surface, maybe a new receptor, maybe a new channel, maybe a new identifying tag, or to release that material from the cell, make a new secretion. All right. As an 8-year-old, you could run around the house for days on end and still smell like a rose. All right. Can my teenager run around the house for days on end and still smell like a rose? No, thanks to proteins, she now has the ability to produce epocrine sweat from her echocrine glands, giving that musky organic sweat that I associated with the stink of her room. Now, think about this. Of the two processes, amino acid based or lipid based, which one's faster?
Amino acid based.
Yeah, if you think about it, amino acid based are basically turning switches on and off. Turn on an enzyme, turn off an enzyme, increase metabolism, decrease metabolism. Clearly, this process takes a lot longer. This is a much, much slower process. But enzymes can be turned on and off quickly. And they can, those effects are going to be short term. If I'm putting a new protein on the surface of the cell, I can make a permanent change to this cell that it never had before. I could put a new receptor and now allow it to be stimulated by a hormone that could never affect it before. So while this is a much, much slower process, it is an incredibly powerful process where we can make long-lasting, in some cases, permanent changes to the cell. Again, truly amazing job drawing this. In fact, I'll save these, share them with you. But let's look at the pretty picture from your textbook. Again, notice here with our lipid-based, here's our 4-carbon chain cholesterol, steroid sneaking right in. Notice forming that complex either in the cytoplasm or in the nucleus itself. When it forms that complex, it's going to bind to the DNA, triggering activation of the gene or triggering transcription to take place, which is going to give us messenger RNA, which then goes to our rough endoplasmic reticulum for translation, goes to our Golgi apparatus for modification, and then becomes a functional protein that can give us a very specific effect. And I think your book's got, so your book doesn't have the inhibitory process, but it does have the pretty picture that goes along with this. At least through the translation part. Doesn't show what happens to the amino acids after that. But especially with Halloween right around the corner, these remind me of candy corns, so it's fun to look at that. All right.
Professor?
Yes.
I have a why question. I don't know if there's an answer, but when that lipid-based hormone enters the cytoplasm, why does it need to be, why does it need to bind to a receptor in order to enter the nucleus?
It's an amazing question. And so I have a great answer for you. Remember, these are hormones that can be distributed throughout the body. And because they are lipid-based, is there anything stopping them from coming inside of the cell?
No.
No, there's nothing to stop them from coming inside the cell. So do we want every steroid hormone telling every cell to make every protein that it tells it to make?
No.
No. The way we control what a hormone affects is by that receptor. By having a receptor, by not just having the hormone, but having that hormone receptor complex, we can make certain that only the cells we want to be affected by the hormone are going to be affected by the hormone. It's even more important for the steroids, for the lipid base, because lipid-based hormones could get inside of every cell. And so we don't want the hormone by itself being able to trigger the change in the DNA, trigger the transcription of the DNA. We want to only be precise with which cells we do that on. The advantage of it coming inside is we don't have to have a second messenger. If it just bound on the outside, then we would need some second messenger on the inside. But since it can easily get inside, we don't have to worry about that. It can just come inside. And then if it's the right cell, it'll find a receptor, bind to that receptor, and then have the change we want it to have. Now, where it binds inside the cell, whether it binds in the cytoplasm or the nucleus, really doesn't matter. Because as we know, nuclei have nuclear pores. So if it binds in the cytoplasm first and then enters, great. If it enters first and then binds, great. It doesn't matter because of those nuclear pores. But we want it into the cell, and then we want that receptor to control what it affects. Did that answer your question?
Yes. Mhm.
Excellent. Any others? All right, excellent. That is, introduction's not the right word, but that is our, for lack of a better word, introduction to hormones and how they're going to influence and how they're going to affect the cell. Now we can start talking about those specific examples. So what we're going to do next, after the break, we'll take a break, and I went a little long here, so we'll take a little bit of an extra break. We are going to start going through the organs. As we go through the organs, we're going to identify the classes, the specific hormones when appropriate, and figure out the targets, effects, and how it's regulated. So if you haven't printed out your cheat sheet for this yet, let's do that so that you have that, or have something where you're taking notes on it, so we can do this after the break. Like I said, I went a little bit long, so we'll take a little bit of a longer break. I'll give you guys a 20-minute break, a real chance to decompress before we have to start back up again. So let's come back at 20 minutes would be 54, and at 2:54 we will restart. And I will start the recording at that time. All right, any other questions before we take our break? All right, take an extra stretch, extra drink, biological break, cry, whatever it is you need to do. Be back in here in 20 minutes. All righty. Any questions on anything we've done so far before we get started? All right, then let's start working our way through the list. We are going to start first talking about the hypophysis, or what is also known as the pituitary gland. Both are appropriate terms for the exact same structure. Where exactly would you find that on this illustration, this chart that's in the classroom? Anybody know where you might find the hypophysis?
In the middle of the brain?
Okay, can you be more precise? Is there any kind of landmark or anything you might be able to use to help us to recognize and identify it on this particular chart?
Is it right under the sella turcica?
Well, I wouldn't say under, but you're getting absolutely warmer. Remember our sella turcica, and let's cheat and make this line bolder, is this U-shaped indentation. A bone feature of what bone again? What bone is that a bone feature of?
Was it the sphenoid?
Yeah, exactly. The sphenoid bone. Absolutely. And someone remind me again, the other name for the sela turcica. Do you remember the other name we gave it?
Saddle.
Well, it is the Turkish saddle. That is what sela turcica means. But remember, the indentation we also called the hypophyseal fossa. Fossa, remembers, an indentation that the pituitary gland or hypophysis sits in. Now, I want you to notice one other not so subtle thing that they've done for us here on our pituitary gland. Notice if you look at the pituitary gland as it sits in that hypophyseal fossa, is that the front of it is more dark in color and the back of it is more light in color. They've given two different colorations to the pituitary gland because there are two distinct regions, and not only two distinct regions, but these two regions are actually made of two different types of tissue. So not surprisingly, they're going to be different in their function and different in their structure. There is one other additional name for the pituitary gland. It is also sometimes referred to as the master endocrine gland. I have to make this smaller to fit it in here. Because it produces well over a dozen different hormones. And as we mentioned earlier, the majority of these hormones are used to control the other glands of the endocrine system. Again, the simplest example, like we talked about before, is we mentioned the thyroid stimulating hormone, which stimulates the thyroid gland to do its job. So for many, many, many years, when this was first discovered, it was believed that this pituitary gland was the command center, was the control of the endocrine system. Now, we have since learned that's not true, and we'll talk about that in just a minute, but it is still considered the master endocrine gland because it produces so many vitally important hormones. It is made up of two parts. We can identify them by their location, anterior and posterior, so the posterior pituitary, or again, we can give it a name based on the type of tissue it is comprised up of. The posterior pituitary is known as the neurohypophysis, and guess what type of tissue the neurohypophysis is comprised of? Exactly, it is comprised of nervous tissue. Now, there is one big important caveat that we have to make about this region. Again, remember, our endocrine glands are glands. Glands, by definition, are specialized cells that make and release a substance. But notice, nervous tissue is not glandular tissue. So as it turns out, this nervous tissue here doesn't actually make its own hormones. Instead, the posterior pituitary, the neurohypophysis, is instead simply responsible for storing and releasing hormones that are made someplace else. We'll talk about where that someplace else is later. This, if you've ever been in the classroom and looked at the nervous models during 430, at the bottom of the brain was this kind of pink M&M kind of dangling in space from there. It is held to the bottom part of the brain, particularly to the hypothalamus, by a thin piece of nervous tissue known as the infundibulum. And in fact, if we cheat and go back to the previous picture. Let's clear this. Let's go back to this. This little stalk-like structure. Let's do it this way. Right here, this stalk-like part right here, that it represents the infundibulum. And notice it attaches to the posterior pituitary, connecting to this region here, which is the hypothalamus. Excellent. The second part of the pituitary gland is in the front, so it's called the anterior pituitary, or also known as the adenohypophysis, and it is made of glandular tissue. So of course, being glandular tissue, it can both make, store, and release its own hormones. Now, with these two regions being different tissue, different regions, they have and produce different hormones. And because we care about such things, they're also regulated differently. So we need to talk about the hormones they produce, what those hormones do, and most importantly, how they are regulated. Of course, before we do that, let's take a little more look, as I promised we would look at the histology of this. This here is our, oh, here's another good place to look at this stuff. So wait, before we look at the histology, let's do one other thing. That's what I want. Come on, chair spring. There we go. Again, here is that Cosumnes River College virtual anatomy lab. Huge advantage of this is that it has those pictures of the models and the materials that are in the classroom, and it has some of the endocrine ones that we are going to be interested in, and you definitely be responsible for on the exam. This one shows some things that we're interested in. Not too worried about that right now or that right now. This is what I wanted to look at. Notice here we see this anatomy on the underside of the brain. We can recognize some of the structures we were just talking about. That infundibulum that is going to connect to the posterior pituitary. It connects up here to the hypothalamus. And if you remember also, we talked about how this pituitary gland sitting in the sella turcica sits right beneath the optic chiasm. Optic chiasm is where you get partial crossing of that optic nerve. We also have here then, change colors. The larger and anterior part is that adenohypophysis, the anterior pituitary. So let's leave this here for a minute and now come back and look at this. Now that we've seen the illustration of it as a whole, we can actually see the histology of it here. Notice, let's start easy. This stalk-like structure, what do you think that is?
Infundibulum.
Infundibulum, and so not surprisingly, it connects being nervous tissue to this posterior part here, which is also nervous tissue. What might, about this as you look at it, indicate to you that it is nervous tissue? What is nervous tissue made up of again?
Ganglia.
Say again?
Ganglia.
Okay, ganglia, and what else?
Neurons,
Neurons. And what are the two main parts of a neuron?
axon, and cell body.
Cell body and axon. And what wraps around most of the axons of a neuron? I'm not here. Myelin sheath, absolutely. All right, I'm going to mute you guys so that somebody can answer that. There we go. You're welcome to unmute yourself to talk after you answer your phone. Myelin sheath, which is made by here in the central nervous system, are oligodendrocytes, where it wraps its plasma membrane, its phospholipids, hundreds of times around the axons, giving it that whitish fatty color. So notice the lighter color is not surprisingly the nervous tissue, whereas the larger portion and the darker stained portion is the glandular tissue. Now notice also with this darker portion, this larger portion, there are kind of three distinct regions to it. The first part, the part that wraps around, actually I'll get rid of this in a second, bring it back in a second. The part that wraps around the infundibulum is known as the pars tuberalis. Notice, secondly, the vast majority of it, the biggest part of it that sticks out here to the front is the pars distalis. But notice if we look closely, this region of the anterior pituitary, the adenohypophysis that is closest to the neurohypophysis, notice even though it's dark, even though it's glandular, even at this very low magnification, it has a different appearance to it. And that is because this is a very unique and very distinct region of the anterior pituitary, right here in the middle next to the posterior pituitary. So that is known as the pars intermedia. Histologically, it is different from the rest of the anterior pituitary. So not surprisingly, it is functionally different as well. In fact, as it turns out, there is only one hormone that can be potentially produced here in the pars intermedia. Any guess at what that one hormone that can only be produced in the pars intermedia might be? If only I had a list of the hormones in front of me, and that list of hormones in front of me included the hormones of the anterior pituitary. And there was something special about one of those hormones of the anterior pituitary that was different from all the other ones on the list.
Is it the melanocyte stimulating hormone?
Yes, how did you possibly know?
Well, you graciously gave us a list that gave us that.
True. And what's special about the melanocyte stimulating hormone?
It has an
Well,
asterisk.
It has an asterisk nested on the list. Now, with a name like melanocyte stimulating hormone, which is only here produced in the pars intermedia, what do you think it does?
Stimulates melanocytes.
Stimulates melanocytes. And what do melanocytes produce again?
Melanin.
Melanin. So what would that do? What does melanin do?
Makes you dark.
Determine the color of your skin.
Changes the color of your skin, exactly. Which is why, as I sit here comfortably in my office chair, if my wife happens to come in, because for the umpteenth time I have forgotten to take out the garbage, luckily I am able to change the color of my skin to blend into the chair so she doesn't recognize me sitting here. Right? That happen?
No.
No, of course not, right? I can't blend into my environment. Are there creatures that are capable of changing the color of their skin to change into their, dynamically change the color of their skin to change into their environment?
Chameleons?
Yes.
Yeah, there are chameleons, and there's some frogs, some other amphibians that are capable of doing these types of things. Moths and other things are able to do it as well. Octopi, another great example. Absolutely.
Cuttlefish.
Yeah, but we can't. That doesn't mean we don't have a hormone that can help to control the amount of pigment we produce, but do we really use hormones to control the pigment of our skin?
No.
No. How do we change how much melanin we produce?
Vitamin D.
Well, vitamin D is activated in the same way, but what activates our melanocytes to produce more melanin? Come on, you're
Exposure.
overthinking this. Say again?
Exposure to the sun.
Yeah, UV radiation. Absolutely. UV radiation is the primary determinant of how much melanin our melanocytes produce. So is this hormone, this melanocyte stimulating hormone, a hormone that we have a lot of in our blood and use on a regular basis?
No.
No. But here in this pars intermedia, we have the potential to produce it. This is not something we normally produce. But we do know we have the potential to produce it. And how might we know we have the potential to produce it?
Because we can get a tan.
Okay, true, but remember, it's the UV radiation that makes us tan. If you think back to the nervous system, like many things we learn about the brain, we learn about them when the brain goes wrong. Young man goes to war, has the back of his head blown off, comes back, can't see, and we know that the back of the brain is where we perceive light. There have been individuals who have gotten tumors in this pars intermedia area, and those tumors can cause uncontrolled production of this melanocyte stimulating hormone. And what happens to their body?
Becomes darker.
Yeah, they get much, much darker in pigmentation as a result of this. Something similar happens, for instance, with Addison's disease. Addison's disease can trigger an overproduction of melanin. So that overproduction of melanin can be an indication of some type of biological problem. So for us, this isn't something we normally use. We have the potential to produce it. It's only in this area. Obviously, let's finish this one off so we don't have to deal with it anymore. What is obviously the target of our melanocyte stimulating hormone?
Melanocytes.
Melanocytes. And what is the effect that they have on melanocytes?
Produce more melanin.
Yeah. For how it's regulated, you can just put a big fat X in there. We're not going to worry about it because it's not something we normally use. So we don't really understand how it's regulated or why it's there or why the potential is there. We know it can be produced. We also know it's something we don't normally use or take advantage of. So obviously how it's regulated will not be a question that I ask you on the exam. All right. Now, I want to go back to that pretty chart from your textbook, because the other thing that it shows really nicely on here is a cross section through the pituitary gland. And notice in this cross section of the pituitary gland, it shows us the anterior pituitary over here, showing the glandular tissue, whereas over here, it shows the nervous tissue. And in particular, these kind of star-shaped cells, these particular glial cells play an important role in, again, storing and releasing our hormones. Remember, our posterior pituitary doesn't produce the hormones. And what do you think these star-shaped cells in the posterior pituitary might be?
Astrocytes.
They do look like astrocytes, absolutely. And we know astrocytes do play a role in helping to regulate ion concentrations and stuff like that. So that's definitely a great guess. We also know they're the most common neuroglial cells, so it's a great guess that way. However, if I happen to have my histology list in front of me, I might be able to come up with another really good guess. I will wait. Pituocytes, exactly. These are the pituocytes. They're neuroglial cells of the posterior pituitary. One last thing, notice with our anterior pituitary, we indeed have all of this glandular area, but notice there's this middle region where the anterior pituitary is right next to the posterior pituitary. And guess what's represented by this kind of different shaped glandular tissue?
Of pars intermediate.
Yeah. Excellent. So this is indeed does a nice representation of that as well. This, like I said, is a chart from your classroom. I guarantee it's going to be on the exam. We have some nice pictures here, and it does almost everything perfectly. There's a couple things it does wonky, but it's still a really, really worthwhile chart, and I guarantee it will be on the exam. All right. Here's actual histology showing basically the same kind of thing. Notice over here, in this case, is the posterior pituitary. And again, we can see the individual cells here, which are the pituocytes in that posterior pituitary. Notice all of this over here is the anterior pituitary, the glandular tissue. And again, we have this funny looking region right here in the center. And that funny looking region right here in the center would again be the pars intermedia. Remember, the pars intermedia is part of the anterior pituitary. It is glandular tissue, but it only can potentially produce one hormone, that melanocyte stimulating hormone. All right. Questions on that? All right. As I mentioned, for many, many years, the pituitary gland was thought to be the command center for the endocrine system. And like I said, it is still called the master endocrine gland. But only about 20 years, which from a scientific standpoint wasn't that long ago. Only about 20 years ago or so, we actually learned that yes, the pituitary gland is the master endocrine gland, but it is actually controlled by something else, the hypothalamus. It's like politics, right? The figurehead in front that everybody sees, right? That's the pituitary gland. That's the president of the United States. Whereas there's that really secret, underground, hidden secret society, right? The the Illuminati or something that is controlling the president from above. Well, it's the same thing with the pituitary gland. Hidden and controlling it from above is the hypothalamus. Now, the hypothalamus is part of the brain, but it's an interesting combination of both neural and endocrine tissue. So it is both a neural and endocrine gland. As we talked about in 430, it plays a huge role on regulating processes in the body, like our thirst, like our hunger, body temperature, limbic system, things along those lines. But what we care about right here is how it controls the endocrine system. And basically, it controls the endocrine system by controlling the pituitary gland. So it controls the pituitary gland, and that allows it to basically be the command center, be the area that gets all the information from the nervous system, from the endocrine system, chemical signals, neural signals, make decisions, and then tell the pituitary gland what to do. We do have one big issue with this, though. The big issue we have with this, let's go ahead and draw this. Here's the hypothalamus. We know it connects to the infundibulum, which connects to the posterior pituitary. Don't like that. A little bigger. There we go. But we also know that we have that glandular tissue here in the front, the anterior pituitary. And what type of tissue is the anterior pituitary again?
Glandular.
Whereas our posterior pituitary is
Neural.
so we have two different structures or two different halves of a structure that are made up of two different types of tissues and need to be controlled two different types of ways. So the way the hypothalamus is going to control the posterior pituitary is going to be different from the way the hypothalamus controls the anterior pituitary. Now, the posterior pituitary is nervous tissue. Guess how we're going to control or regulate the activity of the posterior pituitary? Not your question. It's neural tissue, kind of like the medulla of the adrenal gland was neural tissue. So how did we control that?
Action potentials.
Yeah, by neural control. Exactly. As we just mentioned, there are some pituicytes which are these little neuroglial cells found inside of our posterior pituitary. And as I mentioned, their job is to store and release hormones. But remember, this is nervous tissue. The posterior pituitary, and I'm just going to abbreviate here, PP, does not make its own hormones. Instead, their hormones are produced in the hypothalamus. So up here in our hypothalamus, we have some neurons. These are neurons found in the central nervous system. So of course, these are interneurons, which of course, as you sure remember from 430, means they're multipolar. And basically, their axons travel down the infundibulum to the pituicytes. Here, they release the neurotransmitter, giving the neurotransmitter to the pituicytes to store. And then they also can stimulate the posterior pituitary to release those hormones as needed. Now, just out of curiosity, how many posterior pituitary hormones are you responsible for?
2.
2. Does that mean it only keeps and releases 2?
No.
No, just means that there's only 2 that you're responsible for. What are those 2?
Anti-diuretic and oxytocin.
Yeah, anti-diuretic hormone and oxytocin. So those 2 hormones are produced in the hypothalamus, stored and released from the posterior pituitary. And as you're filling out your chart, how are both anti-diuretic hormone and oxytocin regulated?
Neural via the hypothalamus.
There you go. So one process that we get to use for 2 hormones. Questions on that one? All right. Now, we have a bit of an issue here. Notice the infundibulum just connects to the posterior pituitary. It's nervous tissue and it controls nervous tissue. Remember, in here, we have all these glands. Some really simple glands here. True, one way we could control these is to send an axon into the anterior pituitary to communicate with every single one of these cells to tell them which ones to make. It would be a bit tedious. And the other issue we have is we don't have a direct route because the infundibulum doesn't connect to the anterior pituitary. So how instead are we going to want to regulate the anterior pituitary? Anyone know? Able to guess because of the hormone lists? Able to guess because they looked ahead at the lecture notes?
Is it hormonal?
Yeah. Via hormonal control. Basically, our hypothalamus, let's not use blue. Blue is going to be bad for this. Our hypothalamus I don't like the light blue. I mean the light green. There we go. Produces two main classes of hormones. Releasing hormones and inhibiting hormones. Guess what releasing hormones tell a glandular cell to do?
Release.
Yeah, make and release a hormone. And guess what an inhibiting hormone tells a glandular cell to do? Yeah, stop making and stop releasing a hormone. These are classes of hormones. Oops. So again, we have these releasing hormones. Inhibiting hormones that tell them what to do. So say, for example, let's stick with one that we have talked about already. Let's say, for instance, our hypothalamus makes thyroid stimulating hormone, releasing hormone. What would that tell the anterior pituitary to do?
Release TSH.
Whereas if it makes inhibiting thyroid stimulating hormone, then guess what happens then?
Tells it to make less
Yeah.
or not release any.
All right, see how that would work? So for every hormone of the hypothalamus, and how many hormones of the hypothalamus are you responsible for?
2.
Pardon me, no, sorry, not the hypothalamus, pardon me. How many hormones of the anterior pituitary are you responsible for?
7.
7, absolutely. Although, isn't one of those melanocyte stimulating hormone?
Yeah.
Yeah, so technically 6. For all the other 6, how are those 6 hormones regulated?
Hormonal via the hypothalamus.
Yep, although remember, we want to say the hormone, not where it's coming from. So hormonally via releasing hormones and inhibiting hormones. Now we do have one more issue. Here inside the hypothalamus is the cell that is making all of these hormones. But I need to get those hormones to travel to the anterior pituitary. Well, what's a good way to get hormones to travel where we want them to go?
Veins.
Arteries and veins, blood vessels. Absolutely. However, we know arteries feed into capillary beds, and then capillary beds exchange their materials and feed into venules that typically go back to the heart. Although if I remember correctly, in the digestive system, that wasn't the case. Blood that took on materials in the small intestine didn't go straight back to the heart. It went somewhere else first. Where was that somewhere else it went?
The kidneys.
Wasn't the kidneys. Where did we take that nutrient-rich blood to be processed before we sent it back to the heart?
The portal vein.
Say that again?
The portal vein.
Yeah, the portal vein.
Hepatic. The liver.
Hepatic portal vein. It went to
yeah,
the
sorry,
liver via a portal system. It went to a second capillary in the liver before it went to the heart. And we called that a portal system, where guess what? The exact same thing happens here. There is a portal system that includes a second capillary, so that these hormones that are produced in the hypothalamus can enter into the blood. They can then come out of the blood and they can control the glands of the anterior pituitary, causing these glands to then produce their hormones. We'll use Clara and I used brown. Causing them to produce their hormones, which will then enter into the capillary and then travel throughout the body like all circulating hormones do. So the same way it's the hepatic portal system, here we have a portal system as well. So notice if we look at the pretty pictures, I know I haven't done the best job of showing this, but let's look at the pretty pictures from your textbook. Notice here we have the pituitary or hypophyseal portal system. Notice the hypothalamus is producing, it's releasing and inhibiting hormones, depositing them into a capillary, which is then carried to the anterior pituitary, where it has its effect. And then those glands then release their their hormones into the blood. And whether it's thyroid stimulating hormone or luteinizing hormone or any of these hormones that are produced, then go and travel throughout the body. So just like we had a portal system in our liver, this special communication pathway of blood, we have a portal system for the pituitary as well. So the good news is for all 6 of those, they're going to be regulated hormonally via the releasing and inhibiting hormones of the hypothalamus. As we also mentioned, this only works for the anterior pituitary, the posterior pituitary, an entirely different system. Anti-diuretic hormone and oxytocin are actually produced in the hypothalamus. They are then transported down to the posterior pituitary, where they're stored in the pituocytes. Then when we want the posterior pituitary to release its hormones, the hypothalamus stimulates those cells. They release it into the blood supply, and oxytocin and anti-diuretic hormone are spread throughout the body. So notice for the pituitary gland, like I said, there are 9 total hormones you are responsible for. The 2 for the posterior pituitary are both regulated neurally by the hypothalamus. 6 of the 7 anterior pituitary are regulated hormonally by the releasing and stimulating hormones of the hypothalamus. And then the melanocyte stimulating hormone, we don't care how it's regulated. Well, let's say it this way. It's not that we don't care, it's just not going to be on the test. So you don't care. I'm sure there are people that care, but they're not taking this class. All right. Questions on that?
Professor, so for the releasing and inhibiting hormones, for the targets, that would just be the glandular cells of the pituitary gland?
Yep. So then that's a great question. So then how are they regulated? How are the releasing and inhibiting hormones regulated?
The nervous system.
Yeah, I would say that that's a fine way to do it, just by the nervous system. But again, remember also, it's going to receive chemical signals as well. So it's the top, right? Who knows what controls the Illuminati, right? Well, it's the same thing with the hypothalamus. Who knows what controls it? So there's nothing wrong with saying nervous system. It's broad, generic, but yeah, it's because it needs a broad generic answer because there are hundreds, if not thousands of things that are interacting and causing the hypothalamus to make the decisions that it makes. It's the top, right? So since it's the top, we don't really worry about that because that's a far more complicated process. You can learn about how the hypothalamus is controlled in grad school. We're not going to worry about it here. So yeah, just saying neural control is totally fine. It's a cheat, but we'll take it. All right. Questions on that? All right. Well, as we can see here, here is the list of hormones you were going to be responsible for the pituitary gland. Again, remember, these are not the only ones they produce. They're just the only ones you are being held responsible for. So let's just focus on those. We'll start with the posterior pituitary, 1, because it's easier, and 2, because it's fewer. As we mentioned, posterior pituitary is nervous tissue. So remember, its hormones are actually produced in the hypothalamus and just stored and released by the posterior pituitary. Let's start with the one we're probably most familiar with, anti-diuretic hormone. What is the target of anti-diuretic hormone?
Kidneys?
Kidneys. Excellent. What does it tell the kidneys to do?
Retain more water?
Exactly. Right? It tells the kidneys to hold on to more water. If you think about it, there's basically 3 effects of that. It concentrates your urine. It increases your blood volume, which increases your blood pressure, right? We don't usually think of it in those terms. We think of it more the other way around because things like caffeine inhibit your release of anti-diuretic hormone. So you take that quad shot of Dutch Brothers on your way to work in the morning, and after you're sitting at your desk for half an hour, what happens?
You have to use the bathroom.
Yeah, you have to pee, or alcohol inhibits it. So you've had that third Long Island iced tea, right? And then what happens?
You try hard not to break the seal, but eventually you have to.
Yeah, exactly. And then you're just going all night long. And not only are you peeing all night long, but you're peeing basically clear water, right? Because it is so dilute, right? Because again, you're not holding on to that water. So you're producing a large amount of very dilute urine, right? And don't even get me started on those four locos, right? Alcohol and caffeine. Yeah, nothing but trouble. Excellent. And even though hopefully we've already emphasized this enough, give it to me. How is anti-diuretic hormone regulated?
Neural via the hypothalamus.
There you go. Perfect.
Professor, quick question.
Yep.
This might be a bit off topic, but why does coffee in some people cause a diuretic effect, but also maybe a laxative effect for some people?
So you've got the right idea. I'm not sure I would consider it a laxative effect, but you have the right idea. Caffeine has the ability in some individuals, in many individuals, to basically irritate, binds to receptors that irritates the lining of the digestive system. And so as a result of that, that can stimulate the essentially positive feedback process that can cause defecation to occur.
But that also includes water, right? Like in the digestive system.
So if you are stimulating the digestive system to void prematurely, and that premature voiding typically means that not enough of the water has been reabsorbed, so the stool can be softer or even be, diarrhea-like as a result of that. But it doesn't have to be that way.
So
Got
it
it.
probably just depends on whatever stage you're in.
Yeah, exactly. It has
When you
more to
drink
do
the
with
coffee
timing,
after
the relationship of when it is, right? For many people, when they're having that coffee first thing in the morning, right, the last time they ate was dinner many hours before. So much of the processing has taken place. So it's mostly done. And so it doesn't have to necessarily be have that causality.
Got it. Thank you.
No, it's a great question. So
I had a question.
yes.
So going back to the anterior pituitary, for the targets outside of the melanocyte stimulating hormone, you said you'd accept the glandular cells of pituitary gland.
Well, so you're talking about the hypothalamus. You're not talking about the anterior pituitary. Remember, the releasing and inhibiting hormones, those two classes of hormones that are produced by the hypothalamus, their target is the glandular cells of the anterior pituitary.
Okay, and you said those are all regulated by the hormonally via releasing and inhibiting hormones of the hypothalamus, right?
Yeah.
Okay.
Notice that's two sides of the same coin. The releasing and inhibiting hormones target the anterior pituitary. The anterior pituitary is controlled by the hypothalamus.
Yeah, I was just reading the textbook, and for follicle stimulating hormone, for example, it said the targets were the ovaries and testes.
Okay, but again, we haven't talked about the targets of the anterior pituitary hormones yet.
Okay, I see.
We've only talked about the target. So far we've talked about the targets for 3 hormones. We've talked about the targets for the releasing and inhibiting hormones of the hypothalamus. And we've talked about the target of the anti-diuretic hormone.
Got it.
We mentioned how the anterior pituitary hormones are going to be regulated, but we haven't talked about their targets or what they do yet. Does that make sense?
Yes, thank you.
Excellent. All right, and we're going there next, but we do have one more we need to do first. Our second hormone of the posterior pituitary, oxytocin. This might be another one that many of you are familiar with. What does oxytocin target? No?
Doesn't it have something to do with, I could be so wrong about this, but labor? Like,
You are absolutely 100% not wrong at all. Absolutely. One of the main targets of oxytocin is indeed the uterus. Remember, we talked about positive feedback processes in 430, and one of them involved oxytocin. Baby's head pushes against the cervix. There are stretch receptors in the cervix that stretch in response to the baby, sending a neural signal to the hypothalamus, which then sends a neural signal to the posterior pituitary, telling it to release oxytocin. Oxytocin causes the uterus to contract, pushing baby harder against the cervix, causing a bigger stretch, a bigger signal to the hypothalamus, a bigger signal to the posterior pituitary. More oxytocin is released. So absolutely, you were 100% correct about that. But oxytocin, notice there's an S there, it has a second target. Anyone know what the second target is?
Breasts.
And specifically the mammary glands of the breasts, absolutely. Now, we've already mentioned, the effect on the uterus. The effect on the uterus is uterine contractions. What's the effect of oxytocin on the mammary glands?
The production of milk
Milk production?
or ejection.
Yeah, the milk ejection, I like that. Or milk letdown. Right? So not the production of the milk, but the ejection of the milk, the letdown of the milk. We just talked about the labor process, that positive feedback where it is causing the baby to be expulsed. But one of the many advantages of breastfeeding is that while baby is breastfeeding and again, stimulating the nipple sends a signal to the brain, the hypothalamus. The hypothalamus tells the posterior pituitary to release oxytocin, and two things happen. Our milk lets down so that the baby can drink the milk from the breast, but the uterus also continues to contract, right? The uterus in a female who has never been pregnant is about the size and shape of a pear. When the baby is expulsed, is it still the size and shape of a pear?
no.
No, it is have to expand to accommodate a basketball. So breastfeeding can help mom to get that uterus back to close to, it's never going to be its original size, but it's able to get it close to the size that it was before. And it plays an important role in milk ejection. Of course, anybody who's ever breastfed is aware of this, because we start to get this cognitive learning process where when baby cries, we know it's time to feed them, and then the oxytocin is released and the milk comes down. And then after a couple months of that, mom and dad finally decide to take a break, go to the mall or the movies or something like that. They're at a restaurant, and while we're sitting there at a restaurant, someone's baby at another table cries, and what happens to poor mom?
She leaks milk.
Yeah, exactly. You can have a letdown reaction as a response to that crying that takes place. Absolutely. Now, notice I said a lot of we and stuff in that, but I don't see any male effects here. Does oxytocin, is it produced? Does it affect males?
Isn't it one of the main like happy kind of chemicals? I think it's there's
I
like
like the description as happy. You are very close to being absolutely correct on that. Absolutely. It is a bonding hormone. Not just for women, but also for, not just for men, but also for women as well. Both women and men, oxytocin is a bonding that comes from intimacy. Again, for mom, another one of those advantages of the breastfeeding, the skin-to-skin contact with the baby is not only is mom producing oxytocin to let down the milk, but baby's oxytocin levels go up as well, and it increases the bonding. So males often will do skin-to-skin contact with the babies to help with that. And again, it is an intimacy hormone. It is often sometimes referred to as the cuddle hormone, right? Again, having a quickie with some stranger in the back of a bar isn't going to produce oxytocin. But intimate contact, cuddling, nurturing can cause oxytocin to be produced. Pets, if you make eye-to-eye contact with your dog, both your and your dog's oxytocin levels will rise as a result of that. So it has to do with that bonding, that intimacy, so helping to improve those types of relationships, increase that bonding. All right, and right, let's beat the dead horse. How is oxytocin levels regulated?
Neural via hypothalamus.
There you go. Excellent. See how the game's played?
Yep.
Excellent. Let's switch to the anterior pituitary. Human growth hormone. Well, with a name like human growth hormone, what do you think it does? Or let's say it this way. What cells of the body do you think it targets?
Bones.
Muscle cells.
True. Well, although, again, human growth, don't we just need to grow bones? Do we just need to grow muscles? No, it has the potential to affect most of the cells of the body. But you guys absolutely hit it on the head. While it affects most of the cells of the body, bone, cartilage, and skeletal muscle are all very strongly influenced by human growth hormones. And what does it do?
It grows.
Yeah, it increases metabolism, causing the cells to grow. But there are two other important processes that it does as well. It stimulates protein production, very important for bone, very important for skeletal muscle, especially skeletal muscle building, skeletal muscle repair. And it helps to facilitate the body to be more likely to break down fat to metabolize for energy than sugars. Right? So it helps you to build bigger muscles faster and recover from injuries. Again, if you look at, you can see why this is something that is abused in sports all the time. And again, when it first was being used and was not being used as sophisticatedly, you had people like, like Barry Bonds, people like McGuire, people like Sammy Sosa, who were big, huge, massive behemoths of human beings. Then as things got more sophisticated, you saw some of these scrawny little pitchers who got popped for human growth hormones, right? They were tiny, they were all lithe. The big muscles weren't necessarily helping them. So why were they using it? The repair. I know baseball is a game, but you do anything for 162 days straight during a summer, and that's going to cause tremendous wear and tear on your body. So to be able to recover from that daily grind of going through that, faster muscle repair means that you're able to, respond at your peak levels for a longer period of time, get the bigger contract, and make all that money. Right? So again, a huge advantage to these types of things. Certain types of dwarfism are caused by the epiphyseal plates closing prematurely. So one of the things that they will do with those types of individuals is they can give them human growth hormone, causing the bones to grow longer, faster. Some people can get as many as 3 inches of height added to them in that. Now, I know 3 inches doesn't sound like a whole heck of a lot, but if you're 4'1" versus 4'4", do you think that that's a big difference for those people?
Yes.
Yeah, absolutely. So again, the potential of it is very important. The other problem, though, is that you then have teenagers taking it so that they can get bigger, so they can get stronger. Notice Barry Bonds didn't become 10 feet tall when he started using human growth hormones. Why didn't he get taller?
Because he took it after he finished puberty.
Yeah, he took it after his growth plates closed, most of them. Which are the growth plates that closed the last? Anyone know? Skull, which is why his head grew like 3 times in size. Right? Notice it also stimulates cartilage to ossify. So often many of these people who abused human growth hormones have problems with their hips, problems with their knees, because of that ossification of the cartilage by stimulating that bone. So again, like a lot of things, this is something that is really, really good and positive. And like most good and positive things, they will be abused by people that want to take advantage of it. And how is human growth hormone regulated?
Hormonal via the inhibiting or releasing hormones in the hypothalamus.
Perfect. All right. Questions on this one? Prolactin. What's the target here?
Mammary glands.
Yeah, mammary glands. Absolutely. And what does it tell the mammary glands to do?
Milk production.
Yeah, this is the one that is responsible for making the milk. Right, as we talked about, oxytocin is the one that lets the milk down, allows it to be expressed, but this is the one that actually causes the milk to be produced. All right. Everyone has mammary glands. Males have mammary glands. Females that have never been pregnant have mammary glands, but those mammary glands are immature. They're not capable of producing milk. However, when stimulated during pregnancy by things like human somatomammotropin and prolactin, they mature and they become capable of producing milk. Now, again, this seems to me to be a very female-centric hormone. Do males have and produce prolactin? What do you think?
It is testosterone.
It's a great question. Yes, they do indeed produce prolactin. The full effect of it is not completely understood, but what it appears to do is it does appear to play a role in enhancing testosterone production. So maybe play a role in emphasizing secondary sexual characteristics, emphasizing aggression, libido, things along those lines. And even though it's not here, how is it regulated?
Hormonally via releasing and inhibiting hormones of hypothalamus.
Exactly. And again, remember that's going to be the answer for all of these. Oh, all right. Every test, there's always that alphabet soup term you know you're going to have to spell. And this is it for this one, adrenocorticotropic hormone. But as we've talked about before, there's a lot in a name. What does adreno refer to?
The adrenal gland.
The adrenal gland. If you remember, I think we've talked about this before. The adrenal gland, of course, sits on top of the kidney, not drawn to scale. And like many organs we have talked about, it has a chewing nougat center and a candy-coated shell. What do we call the chewing nougat center?
Medulla.
Excellent. And what do we call the candy-coated outer region? Cortex. Absolutely. Excellent. So notice adrenocortico refers to the cortex of the adrenal gland, And anybody know what tropic refers to? Growth, activity. So basically, this hormone controls the activity of the cortex of the adrenal gland. So, not surprisingly, that's what the name is, and that's what it does. It targets the cortex of the adrenal gland. Now, the adrenal gland is an endocrine gland that also produces lots of hormones. So we'll have to talk about that a little bit more later. But in general, adrenocorticotropic hormone stimulates the production and release of a specific class of hormones called glucocorticoids. Corticoid means that it is a steroid hormone that is produced by the cortex of the adrenal gland, because remember, the cortex of the adrenal gland is one of the places where we make our steroid hormones. And with a name like glucocorticoids, what do you think it plays a role in?
Glucose regulation.
Yeah, regulating glucose metabolism. Now, again, insulin helps us to regulate glucose levels in our blood, blah, blah, blah. In this case, we're talking about while dealing with stress in a stress response. So this is going to help us to regulate glucose levels during a stress response. So we're not talking about normal levels day to day. We're talking about regulating glucose metabolism when we are dealing with the stress, when that bear with an axe walks into the room.
Now, I know ACTH also stimulates cortisol production and release. Is that just not going to be tested?
Remember, for our classes of hormones, we're going to need to know specific types. And what do you think the most common glucocorticoid is?
Cortisol.
There you go. So when we get to the adrenal gland, we will absolutely positively talk about that.
Okay, thanks.
Yep. And yes, you absolutely positively are responsible for that. But not there yet. We just know that this is what controls it. We don't know what these things do yet. We haven't talked about them yet, but we'll talk about that more later. All right. Two more of the anterior pituitary. Those are our gonadotropins. As the name gonadotropin, tropin again, growth, development of. Gonado are, of course, our gonads. So those are the ovaries and the testes. And there are two gonadotropins that are produced by the anterior pituitary. The first is follicle stimulating hormone. And of course, as we just hinted at from the name, the targets are going to be the ovaries and the testes. However, is it going to do the same thing to the ovaries and the testes?
No.
No, of course not. So in the ovaries, in females, it does two primary things. It causes the production, again, of a class of hormones called estrogens. The most common specific one is estradiol in the ovary. And the other thing that follicle stimulating hormone does is it triggers the maturation of the egg, or what is appropriately referred to as the oocyte. Notice I said maturation and not making. Why isn't it responsible for making the egg?
Because when a female is born, she already has all of the eggs.
Absolutely. Females, being the overachievers that they already are, they are already working on the grandkids while mom is carrying them in utero. So Amanda's 100% correct. When a female is born, she is actually born with all the eggs she will ever have for her entire life. Right? And then her job will just be then to mature them and release them, and then all the fun that comes along with that. Males, on the other hand, are not born with any sperm. They don't start producing sperm until puberty. But once puberty hits, what happens then?
And they make it all the time.
Yeah, exactly. They basically are sperm machines, making sperm 24/7 from puberty till when?
Till death.
Yeah, pretty much, right? Males stop producing sperm about 15 minutes after they die. So follicle stimulating hormone stimulates that sperm production in males. The second gonadotropin is luteinizing hormone. Again, it's a gonadotropin, so it's targeting the ovaries and the testes. And again, the function is going to be different in males and females. In females, and again, this is a little bit of a cheat, but I will show you why in just a second. It produces a class of hormones called progestins. And the most common progestin is progesterone. This hormone is also the magical step that triggers ovulation, the release of the egg so that fertilization can take place. In males, luteinizing hormone triggers a class of hormones to be produced, and those are androgens. And the most common androgen is testosterone. So notice here, I didn't use the class names for the hormones, but that's because progesterone and testosterone rhyme. And so it helps me to remember the two hormones that luteinizing hormone produces. So yes, technically it's the whole class of androgens. Yes, technically it's the whole class of progestins. But progesterone, testosterone helps me to remember them. Questions on that? And since I haven't said it in a while, let's say it again. How's it regulated?
Hormonally via the releasing and inhibiting hormones of the hypothalamus.
There you go. Music to my ears. Excellent. And that brings us to thyroid stimulating hormone. Based on the name, guess what its target is?
The thyroid.
Thyroid gland. Guess what its effect is?
Stimulates the thyroid to, wait.
Yeah.
Am I wrong on that? To release TSH?
Nope.
T3 and T4.
Exactly. You're right. It stimulates the thyroid gland to produce and release its own hormones, the thyroid hormones, and in particular T3 and T4. And let's beat the horse to death. How's it regulated? Hormonally via releasing and inhibiting hormones of the hypothalamus. Beautiful. Excellent, excellent, excellent. All righty. So there you go. The good news, we've got most of the heavy lifting done. There are a couple more that's going to take a little bit more time to work our way through. And obviously with the gonads, we are just going to touch on the surface of what they do. When we get to the reproductive system, we'll talk about those in much, much more depth. But this, what we've done here, is pretty much what we're going to do for the rest of the class on Thursday. Identify a gland, identify the hormones, and talk about some of the anatomy, talk about some of the histology, and then talk about their targets, effects, and how they're regulated. All right. And we finished a few minutes early. Questions on any of that? All right. Excellent. You have a couple days to decompress. I will see you on Thursday, and we will finish off the rest of this. So you guys have a good day and a half off, and I will see you at noon on Thursday.
Thank you.
All right.